Cajal, and we said that these are like pacemaker cells, very similar to the Purkinje cells in the heart. And what happens is that in each region of the gut, you've got a set of interstitial cells of Cajal that generate the rhythm for that particular region. Remember, we said that in the stomach, the antrum, the contractions are more, the slow waves are more. And as you get to the ileum, it increases, at least in the duodenum, the rate increases to about 12 per minute. And then it goes decreasing until you get to the end of the ileum. So that's how these cells control the motility in a particular region. And what we said is that when you have loss of these cells, for whatever reason, you can get abnormal gut motility. And this slide shows you, if you read the names, they're just talking about distension. It's usually something to do with the wall. So you can get gastric distension because you can't get contraction and thus releasing of the gastric contents into the duodenum. You can have abnormal small bowel motility, whether there's obstruction or not. Inflammatory bowel disease, the chronic idiopathic intestinal pseudo-obstruction. Even tumors, you can get tumors of the gastrointestinal tract, either affecting the autonomic nervous system or the stroma. And in the esophagus, what you'll get is achalasia. And in achalasia, because remember the esophagus, I don't know, I'm sure this we'll talk about later. The top 1/3 of the esophagus is comprised of skeletal muscle. The bottom 1/3 is comprised of smooth muscle. But in the middle, you have a mixture of smooth and skeletal. And sometimes that is where you're going to get difficulty, because remember, this is where the enteric nervous system is going to start, and you can get a lack of communication between the skeletal and the smooth. So what happens is that as you swallow the bolus, or as more food comes down, you get distension in the esophagus. But then you can't get the reflex contraction because of damage to the ICC or the enteric nerves. Anyway, we'll discuss that when we come to look at motility in the different regions. So that's achalasia. You also get intestinal obstruction with hypertrophy, so which means that the muscle coat is going to hypertrophy in the small intestine. You can also get Hirschsprung's disease. Now, Hirschsprung's disease is also known as congenital megacolon. And what happens is that during development, the neurocrest cells, as they migrate caudally, sometimes do not get to the sigmoid colon or even the furthest part, the distal part of the colon. And so what happens is that the digestive system develops, but you have a lack of enteric nervous system in that region. So when the baby is born, after about 2-3 days, they notice that she doesn't pass any stool. So, and then when you examine the abdomen, it just gets distended. So you're getting motility along the gut, and it's distending the gut wall, but you don't get the associated contraction, which is going to help to propel the contents. So it's usually discovered very early in life. Fortunately, once it's detected, once you identify it, you can actually remove the part of the colon where there is no ganglia. And then you can connect the two cut ends, and usually it will heal if it's not a very vast region, and the person will develop grow and develop afterwards quite normally. Okay, so you have all manner of forms of pseudo obstruction. You can also get slow transit constipation. So this constipation is occurring because the contents are being moved too slowly, maybe because of a lack of coordination, because the nerves, between the nerves and the blood vessel, the nerves and the skeletal muscle, sorry, the nerves and the smooth muscle. You can also get anorectal malformations, which will imply a congenital problem during development. So it's combined. It's a failure of the enteric nervous system to develop and to coordinate with the smooth muscle. So these are just examples of what can happen. So they're very important in maintaining rhythmic movement or rhythmic contractions along the entire length of the gut, from the esophagus, bottom 1/3 of the esophagus, all the way to the anus. Now, this is just a picture I think you can get. I think it's in your book. It just shows you the action potentials and the basic electrical rhythm. And because I've enlarged it, that's why it's blurred, but you can have a look at it at a later time. So basically what we're saying is the motor activity of the gut, you have contraction of the smooth muscle, which is synchronized. It's a synchronized contraction and relaxation. So it's not just contraction alone or relaxation again. It is synchronized in such a manner, as we said, as to propel the contents. And remember, when this smooth muscle of the gut is contracting, remember we said the outer longitudinal contracts and shortens the gut, the inner circular layer contracts and reduces the lumen. So that combined movement of the contraction and relaxation of those two muscle coats is what is going to facilitate the propulsion, the movement of content from one area to the next. Now, the amplitude and the frequency of slow waves are altered by input from extrinsic nerves, other intrinsic nerves, okay, the enteric nervous system, and also the circulating hormones, which we mentioned earlier. To summarize or to just generalize, basically the sympathetic, when stimulated, the sympathetic nervous system, you get a decrease in the amplitude of the slow waves on the gut, in the gut, and sometimes it can even abolish the slow waves altogether. Whereas when you stimulate the parasympathetics, generally what you're going to get is an increase in the amplitude. So they work opposite, the effects are opposite to each other. When the threshold is achieved by the peak of a slow wave, then an action potential is fired. And you get the entry of calcium ions, which then initiate, the calcium ions are entering into the muscle, and then you get a contraction. The greater the number of action potentials occurring at the peak of a slow wave, the more intense the contraction of the smooth muscle. So you can see that the action potential, it's not just the presence of an action potential that is going to result in contraction of the smooth muscle. Now, we go on to now the applied or almost applied aspect. And what is important for you to understand is peristalsis, because the movement or any movement, if you ask anybody movement in the gut, the first thing that comes to the mind is peristalsis. So we need to understand what peristalsis is. So the next couple of slides will explain that physiologically to you. So in this diagram, you can see the bolus coming from on top here. And when it passes, or when the bolus or the food content, or the luminal content passes to a certain area, it's going to cause a distension of the wall, okay? And that distension of the wall is the trigger, or is what is going to trigger the reflex that is called peristalsis, because peristalsis is a reflex, okay? So you can see that what's going to happen now is you're going to get relaxation ahead of the bolus and contraction behind the bolus. So when you squeeze behind the bolus, then you're able to push the content ahead, and then ahead of that distension, you get relaxation, so that when this content is pushed, it has somewhere to go. So if we were now to look at this and describe it more concretely, we can say that peristalsis is the progressive contraction of successive sections of circular muscle. Okay, now this muscle is going to, and smooth muscle is going to be in the gut, in the circular layer, and also in the ducts. And that is how you're able to even remove the contents of the duct or the secretions, and they're able to be propelled along the gut, along the gland of the gut. Now, because this is a reflex. Okay, basically what you're going to have, and I think I'll show this in a slide later on, so let me explain it here first. When you have distension of the gut wall because of accumulation of luminal content, you're going to get two responses from that one distension. Ahead of the bolus, you're going to get relaxation, which means that the lumen will widen, whereas behind that distension, you're going to get contraction, a narrowing of the lumen so that you can squeeze the contents forward. So you have one stimulus, which is the distension of the wall, but you're going to get two responses. Ahead of the bolus, let's call it a bolus for now. Ahead of the bolus, relaxation. Behind the bolus, contraction. So peristalsis is a reflex. And because of that, it means that every time you get distension, you'll always get contraction behind the distension, and ahead of the distension, you get relaxation. So you're going to get a one-way movement. The contents will always be moved forwards. They'll always be moved from the mouth end towards the anus. So we say that the gut is polarized. So you have distension of the wall, causes reflex relaxation ahead of the bolus, is what I think I've described. And you get the release, the stretch releases serotonin, which activates sensory neurons, which then activate the myenteric plexus. But behind the bolus, the cholinergic nerves are going to release substance P and acetylcholine, and thus cause muscle contraction. Okay, so distal to the bolus, you're going to get the activation of neurons that secrete nitrous oxide, VIP, and ATP, producing relaxation in front of the bolus, and then you're going to get contraction behind. So this movement forwards from the mouth towards the anus is known as the law of the gut, and it's what results in the one-way movement. So peristalsis will always move substances forward. So I've just written it here again and said peristalsis is a reflex. Now, I know people talk about anti-peristalsis. And I think when they talk about anti-peristalsis, they're talking about movement in the opposite direction of what you expect when peristalsis happens. So for example, when you have vomiting, because I know you're going to ask that, yeah? But when you vomit, you have vomiting centers in the central nervous system, which can override what is happening along the gut wall. So what they do is they're the ones that are going to cause a reversal of the movement of contents from the stomach, usually from the duodenum, maybe part of the jejunum, back to the stomach and back into the oral cavity and out. Okay, and so it's a protective mechanism, usually to either empty the gut, maybe because it's been too laden or with too much caloric food, or you've eaten something that is bad, or there's too much toxin in the luminal content. So you need to evacuate the contents as soon as possible, because usually whatever you ingest into the mouth will end up being digested. It will be processed by the digestive system until it is expelled at the anal end. However, if there's something like we said, for example, there's a reason for you to want to void the contents as quickly as possible, in the upper half, that's what I'm saying, from the jejunum upwards, that you will empty through the mouth via vomiting. However, if you have an irritant or too much secretions and it's lower in the gut, in the ileum, going towards the colon, then what will happen is that you'll get an increase in activity, in motor activity, and you'll end up with diarrhea as a way of trying to rapidly evacuate whatever's in the content, in the lumen of the gut. But you cannot empty gut contents from the colon all the way back out through the mouth. That's what we're trying to tell you. You'll mess up everything, including the gut microbes. You'll find microbes now in the mouth, which should have been in the colon, which is not good. So this is what I was saying, that you have one stimulus, which is the distension caused by luminal contents accumulating in the gut, but then you're going to get two responses. Ahead of the distension, relaxation, and behind you're going to get contraction. So this is also taken from one of your textbooks, And this just shows you the same thing that we have seen, so that you can see at any point in time, wherever you have a distension, you're going to get this reflex contraction and reflex dilatation or relaxation, which will propel. And then when the contents go to the new place, they cause a new distension, which is going to cause a new peristaltic reflex. And that is how the contents are going to move from one point to the next. Now, the reflex, the peristaltic reflex, can be evoked by stroking the intestine or even by circumferential stretch, okay? Now, circumferential stretch is what happens when you get accumulation of luminal contents, because you're distending, you're increasing the circumference, okay? But stroking the intestine, this can happen, for example, if you're doing surgery of the abdomen. So even if you're touching it from the outside, stroking it, that can also cause peristaltic reflex to be evoked. And usually what you're going to get is descending relaxation, caudal or towards the anal end, and ascending contraction or on the oral side of the stimulus. So I'm just saying the same thing again and again. This diagram, I think, just says the same thing, okay? Now, if all that happened in the gut was always peristalsis, okay? Distension, then relaxation ahead, contraction behind. If that was all that happened along the gut all the time, you'd end up with diarrhea. So what happens is that these patterns of contraction change from region to region, okay? So you'll either end up with diarrhea, or you'll end up with the luminal contents transiting through the ileum and through the jejunum so fast that there's not enough time for the final digestion and absorption to take place, okay? Now remember, the final digestion, which we shall discuss next year, and the absorption is taking place at the cellular level, and the contents have got to pass through the mucosal barrier or pass through the epithelial barrier in order for them to get into the bloodstream and then be circulated around the body. So you need time for that. So you'll find that you have variations in these patterns which facilitate that. So you're going to get different types of peristaltic activity occurring from region to region. Now, first of all, you have two basic types. You've got a tonic contraction and a rhythmic phasic contraction, okay? Now, the rhythmic phasic contractions are the ones that you normally associate with peristalsis. They're the ones you're also going to see when you're in the gut practical, and you're going to see the duodenum, the jejunum actually contracting and relaxation. And if you observe, you can see that you can get a ring of contraction, which then relaxes. A ring of contraction, and then it relaxes. So you can imagine if the lumen was full, you'd be able to squeeze the food along, propel it from one area to the next. When it comes to tonic contractions, tonic contractions occur in the gut. So imagine you've got a small amount of ileum, let's say a small part of the small intestine. What will happen, and this you can see with your naked eye, is that you will notice a contraction occurring in a portion of that gut, but the contraction is ever so slight, and then it relaxes. So basically what we're saying is that you get the contraction, you can see the muscles in the gut wall actually relaxing, but you don't get a change in the circumference or in the luminal dimension. So the lumen doesn't get smaller. So what is happening is that you're getting a tightening of the wall, of the muscles in the wall, but you're not changing the luminal diameter. That's what we're calling a tonic contraction. And this is useful because you have lymphatics and venous drainage, and you also, the blood supply in the gut. And remember, while digestion and motility and even absorption are taking place, you require a very good circulation. And as we said, you only have one heart, which is in the thoracic cavity. If the intestines are engorged and there's all this activity taking place, how are you going to propel the blood so that it doesn't become static in a particular area of the gut? So these tonic contractions help to move not only the blood or the venous drainage or venous blood along the gut wall, it also helps move the lymphatic the lymphatic fluid, because what you'll notice is that, or I'm sure you're aware, is that basically the byproducts of carbohydrate and protein will end up in the blood, in the venous blood, then they're carried by the hepatic portal vein. But the fat, digestions of fat, digestive end products of fat will end up being directed into the lacteals, which then join the lymphatic system. So that's one way in which you can reduce the amount of fat, which is actually going to be found in the bloodstream while it's circulating around the body. Okay, so what kind of contractions do we have? Propulsive, that's what we described when we were discussing peristalsis. That is basically the propulsive. It moves the luminal content from one area to the next. Then you have a second type, which is known as segmentation. Segmentation takes place in the small intestine. And what it does, instead of having one particular distension and only one response to that, so that you move the content from one area to the next ahead, you get one or two or three, or even more, multiple points of contraction, which occur in response to the distension. So, and then what they do is they contract and relax. And because they're it's at intervals, what happens, you don't get a smooth moving of the luminal content from one area to the next. In fact, depending on how rapid the relaxation and contraction takes place, you find that the luminal content at that particular point in the intestines can be moved forwards and backwards, because you're going to get the contraction and the relaxation occurring at various points. So what this does is it prevents the luminal content from rapidly moving from one area to the next. Let's say, for example, from the jejunum to the ileum. And by keeping it in the jejunum longer, or in that part of the small intestine longer, then the luminal content has got a greater chance of interacting with the villi and the microvilli in the epithelial cells, on the enterocytes, and so absorption and contraction can take place. So you've got to remember that enzymatic breakdown requires time. Okay, enzymes require time. You can't put them on and off like the switch of a light. So you require time. So by keeping the luminal content in a particular area longer, then you facilitate the final digestion and also the absorption, so that substances can then subsequently move from the lumen and end up into the bloodstream in the connective tissue. The tonic contractions I have already discussed, so they'll be found all along. So remember, propulsive is basically your basic peristaltic reflex. Segmentation is like a chopping up, and relaxation of a small area of the small intestine in an attempt to keep the luminal contents in that area longer to facilitate the functions of the gut. Tonic contractions we've already talked about. Now, if you've ever had the pleasure of slaughtering a goat or a chicken, you'll notice that even after the animal has died and been bled, you will still see writhing movements of the intestine. Because remember, we said the smooth muscle can actually, so long as you have enough blood supply, enough oxygen, and there's not enough buildup of metabolites to slow things down, it can stay for a longer time. So the intestines, I'm calling them intestines now. Okay, the small bowel, the large bowel, they also slide on top of one another, because remember, they're coiled inside the abdomen. So as their content, as their smooth muscle is contracting and relaxing, the coils slide on top of each other, which is why you have your peritonic fluid, the fluid of the peritoneum. And because you have the serosa, and this fluid prevents the serosa from different segments from adhering to one another. So that even as you're getting motility, movement taking place and mixing within a particular region, they slide over one another, and that's what provides the pendulous movements. Remember also that your abdominal organs are suspended via mesenteries. So they don't just move in one place. They're actually contained in the quadrant, in the various quadrants. They don't go floating around all over the place. So your liver doesn't float down into the pelvic region. No, it is held in place. Okay, so the pendulous movement also helps in the movement as the content are moving along. So how fast do the peristaltic waves move? Figures have been given of between 2 to 25 centimeters per second, and that is considered normal. So you can see it can be very slow or very fast depending on what the contents of the lumen are. The more fatty or the more calorific, then the slower the peristaltic waves will be. Other types of contraction, okay? So remember, segmentation is only found in the small intestine to facilitate mixing, chemical digestion, and absorption by slowing down the rate at which the contents are going to travel. Now, in this large intestine, in the colon, in the colon, you have what is known as the mass movement, okay, at the bottom of the slide. Now, remember we said that the small intestine has got a very narrow lumen compared with the colon. So, and also in the colon, water is being absorbed more than is being secreted into the lumen. So the contents are drying out so that you're converting the luminal contents from semi-fluid to semi-solid as more and more water is absorbed. That means that the muscle is going to have to generate more force, so it has to be stronger to be able to push this semi-solid material to move it from one area to the next. And this is where we're telling you that in the colon, you've got the arrangement of the longitudinal muscles so that you have 3 bands along the length of the colon. So the circular muscle fibers will go around the circumference. The longitudinal fibers, instead of just running parallel like in a spaghetti or a piece of pasta, what happens is that they're bunched together so that you have them forming a band, like you have like a belt on top. And because they're together and they're going to contract at the same time, they're going to be able to generate more force to be able to move this semi-solid material along the length of the colon, okay? At the same time, you can also have a large part, for example, the whole of the ascending colon or the whole of the transverse colon can contract at the same time, so that when it contracts, then that way it's able to propel a larger volume of semi-solid content. So mass movement is only found in the colon. You also have another type of movement, which is known as the mean migratory complex or the migratory motor complex. Now, usually it's abbreviated as MMC. And what happens, it's migratory because you get a ring of contraction that starts from the pyloric part of the stomach. It travels distally, that ring of contraction. If you're able to observe, if you're able to open the abdomen, you'd see that ring of contraction traveling across, going from one area to the next. It occurs every 90 minutes or so, so every 1.5 hours. And it travels at a rate of 5 centimeters per minute. What is important about this migratory motor complex is that it only occurs in between the periods of eating. Okay, that's why it's called the interdigestive period. So let's say you've eaten breakfast at 6 and you're going to eat again at 1. Okay, in between, from around 8 o'clock to 12:30 or until you start eating, you can have this a ring of contraction that occurs every 90 minutes. And what it does, because it's a ring that is moving from one area to the next, so you have the contraction and then it relaxes, then the next cells contract and then relax, so that when you're observing from outside, it looks like the ring of contraction is actually traveling from one area to the next. So it milks out, it cleans out the inside, the luminal contents, so that you don't get stasis. Because even if, let's say you're not eating, let's say you're on a fast, the secretions never come down to zero. You'll always have a basal secretion rate. But if those secretions lie in that part of the stomach or the abdomen and they're stagnant, you'll get a buildup of microbes in there because the temperature is interesting, it's ideal. You may have some luminal food content. So you need to be able to clean that out. Also, any residual food or luminal content that was not emptied, for example, like when the stomach is emptying into the duodenum, or when the content is going into the jejunum or into the ileum, all that will be pushed along. And it stops, this mean migratory motor complex stops immediately on the ingestion of food. So the minute you start eating, the mean migratory complex disappears. It doesn't occur again until you finish that meal, and then it starts again. Now, it's also known, and if you look in your textbooks, this mean migratory motor complex is known as the housekeeper of the stomach. So it's how the stomach cleans itself. Okay, the example I would give people, which I think probably makes sense, is I'm sure you remember when you were younger, maybe eating fruits or swallowing seeds. And I'm talking about seeds like plum seeds that you haven't chewed. I'm trying to think, okay, let's say even passion fruit seeds, because I know there are some people who will not swallow a passion fruit seed without crushing it, whereas others will just swallow it. But if you swallow it, that seed is going to be too big to be passed through the pylorus at the time of gastric emptying. So why is it that we don't have accumulation of this solid matter in our stomach after all of these years? Those of you who've even swallowed, or children swallow buttons, they swallow beads, they swallow so many things. Why don't they just fill up in the stomach or get stuck in one part of the intestine? Usually, if you swallow something that is rounded or spherical, then you're okay, because what will happen, it can peruse, it can go through the digestive system without lacerating the mucous membrane, because if you have any lacerations, that will be painful, that will cause havoc. So what happens is that when this mean migratory complex is traveling along, you get this constriction, narrowing of the lumen, it pushes whatever secretions and leftover food remnants are there. And when it comes to where you have, like, for example, a sphincter, for example, the pyloric sphincter or the ileocecal sphincter, when it is passing through that sphincter, the migratory complex causes the sphincter to open, to relax, so that substances can then pass through. So that is how it's able to clear and to clean whatever's left over in the stomach. And as you can see now, that if that is what the purpose of this migratory motor complex is, then it is very wrong of us to keep nibbling throughout the day, because you don't give this migratory motor complex time enough to remove and clear parts of the digestive system. Another thing that you could do, and these experiments, remember, we're not teaching you this information for your patients only. You yourself has got a digestive system, and we're teaching you normal physiology. So if you don't eat for a day or for 2 days, and let's say you're just taking water only, and water doesn't need to be digested, so it will just be released. It will just be, the stomach will just release it into the intestine, it will continue flowing. But if you fast, you give your migratory motor complex ample time to perform this task of cleaning the luminal content or the luminal space. And if you want to, you can actually do this as an experiment you do on yourself. You don't need to tell us, but you can just observe it for yourself. If you don't eat today, tomorrow, usually by around 24 hours, you will go to the loo for a long call, and you'll be shocked because you haven't been eating anything, but you produce a lot. Okay, that is now the digestive system emptying itself. So from that, you can see that people who are continually nibbling, never fasting, never going for long periods without eating, never give the digestive system a chance to empty, which means that every time that you eat, whatever you've just freshly ingested comes and mixes with what was already there, with old luminal content, which is not a good thing. So anyway, we can discuss that another day. But I just want you to know that there's different patterns of contraction. So peristalsis is the generic name. It's the general thing that's going to happen. The distension of the wall, resulting in relaxation ahead and contraction behind. But you have different patterns of contraction depending on which region of the gut you're in. Now, when you're in the stomach, as soon as you ingest food, the pylorus closes tightly. And then your stomach churns the food, and the food, once it gets to the pylorus, because it can't continue into the duodenum, and the pylorus is highly muscular, the pressure that is created will push the food back into the fundus so that the stomach acts like a grinder, like a mill. And it will do that until the chyme is fine enough to be squirted through the pylorus. So the pylorus doesn't open wide and just drain everything into the duodenum, because that way you'll overwhelm the duodenal mucosa by producing too much acid. And so what I'm saying is that different regions, you've got different types of motility, and yet it's all based on peristalsis. Now, I tried looking for a diagram to show you segmentation, so I shall refer you back to the books, but I just wanted to compare propulsion and segmentation. So the top two diagrams at the top show you propulsion, which is where you have distension, just one area of distension, causing contraction behind, okay, the dark area here, that is the food content or the luminal content. And then you're going to get relaxation ahead so that when this contraction occurs behind, it's able to push this content ahead. And when it goes ahead, it then creates a new distension, which then repeats the cycle again and again. And what I was saying is that if that was all that happened in the gut, you would just end up with the luminal content being propelled for, when I say forwards, I'm meaning from the stomach towards the anus. There would not be enough time for chemical digestion and absorption to take place. So for segmentation, this shows you that instead of having one point where you have the contraction occurs in more than one area. So you can see here that you've got 1-2 points of contraction. So if you look at the content, which is colored in yellow, green, and almost purple on this side, you can see that because of the areas which are contracted on this top figure here will relax in the next second or so. The areas that were relaxed will then contract. But because it's in several instances, if you're in the lumen, you can end up dancing to and fro. The content will be pushed forward, pushed back, pushed forward, pushed back. So that is what segmentation is all about. And I guess it was called segmentation because when you're outside of the intestine, looking at the intestine, you'll see like the contractions create like little tiny segments. And then those segments where there was dilatation, they contract, and what was contracted becomes dilated. So that is how you're going to get food. I'm calling it food. It's no longer food now. Luminal content, chyme, okay? Being moved and mixed and coming, this facilitates it coming into closer contact or coming into direct contact with the mucosal wall. And this is what is going to facilitate the functions of the gut. Now, okay, that's the end of this one. I'll share this PowerPoint presentation with you. I want to share them, maybe about 2 or 3 of them as soon as I finish. And these, okay, some of them are quite old because now the enteric nervous system has been accepted and established. But there's also another paper here on the autonomic nervous system. So don't be afraid of the paper. Just read the introduction, because that's what's going to give you the literature review and help you build up on. You don't have to read about the rest of the experiment. You can ignore that. There's also another paper here on glutamine metabolism, which I think would also be useful for you. So that is the end of that one, which is now the motility, how motility occurs. and I'll leave you to read more of this, or to, when you're reading the textbook, to be able to connect the dots and be able to understand this better. Now, let's move on to the next one, because I've just remembered. Next week, there's no class, is there? Next week is your exam week, is it not? Mid-semester?
Yes, next week we have our cards, although I've seen in the schedule
There's some lectures.
Yeah, they've still given you lectures on Wednesday and also Thursday, actually.
Yeah, now there's so many things that are really clashing. So other than clashing with other lectures, next week, because I thought when I saw the exam timetable, I thought it was a non-teaching week. Next week I shall be away because Moy University has exams, so I have to go and participate in those exams. And I was looking at my schedule, and I don't think I will be free. Definitely not Wednesday. So if you can find someone to use that Wednesday afternoon, that would be good. Or if you want to use it to revise, that's okay. And then I'm wondering Thursday, oh yeah, no, Thursday is also in the morning. Thursday is also, I'm also occupied in the morning. So next week I will not be available. So if you want to, you can maybe organize with someone else or make up or something. And then we shall meet again when you come back in January. Is that okay?
Yes, that's fine.
So what I will do, let me, okay, there's today and there's tomorrow. So let me go through as many PowerPoint presentations, and then I'll give them all to you and you can go through them in the holidays. Okay, so let's go. So the next one now going on from this is, remember we talked about peptides, gut peptides, and how they're also used in the regulation and control of gut function. So they bundle them together and they call it humoral control. But remember, some of them act as hormones, some of them act in a paracrine manner, and others are neurosecretions, or they're secreted from the nerves. So back to the gut, because to this saying, because Hippocrates is the first one who said, let food be your medicine and medicine be your food. Now, usually these days we just eat to please our tongue or to quieten our stomach. We don't really think about what we're throwing into our bodies and what effects it's going to have on us until you wake up one day and you're told you're obese or you've got too much cholesterol or things like that. But this is something that has been creeping up because of the dietary habits that you have. So what they were saying is, and I guess even if you talk to people who are more traditional, less westernized, they didn't eat for pleasure. You eat to build up your body. So you eat, you take in the nutrients that are required by your body for repair and regeneration and for growth. So that's why they say, let food be your medicine and medicine be your food, excuse me. So what we're saying is that some things can be cured. Okay, if you eat wrongly and the wrong combinations, you can end up being sick. And if you correct your eating habits, it can sometimes undo what had gone wrong, depending on how long it had gone. Obviously, if it's a habit that's been there for 2, 3, 4 years, which has caused even anatomical changes, then that would be difficult. But physiologically, if you just listen to your body, you can eat. I mean, many of us are like, now you're reading for exams, you're going to be busy, you're not going to be eating at the right time, you're going to be snacking, living on junk food. Yes, but if, let's say, for example, when you finish next week, you go home and you say, okay, I'm going to eat a proper diet, balanced diet, ideal in all the ways that people say that it should be, you will find that your body will recover and recuperate and come back to normal. So what we're saying is that what you eat is very, very important, not just in providing calories, but in maintaining the health of all your systems. Because other than through your digestive system, through the mucous membrane of the gut, the other way that substances can get into the body is either through the skin. For example, when we apply ointments or substances, if you apply them on the skin, they can be absorbed, which says a lot about people who have who go to the hairdressers and do perms and apply bleaching agents, those things get absorbed, they end up in your bloodstream. So you can get substances going in naturally through your skin, through the lining of your mucous membrane in your gut, and also if you inhale chemicals in the air, they will also dissolve in the mucous membrane and of the respiratory tree and be absorbed. And by the way, we do use those roots as doctors to apply medicaments to people. For example, your inhalers, or even what's the other thing? Suppositories, where you give things per rectum. If the person is comatose, they can't swallow, or the medication is not palatable, it's not tasty, or it cannot form a capsule or a tablet, you can actually put it into the rectum, where it will dissolve in the mucous membrane, in the lining the membrane, and it will be absorbed and go into the bloodstream. So you just need to remember that. Now, when it comes to the gut peptides, we have different entities, and the gut motility can be generated and modulated by local neurohormonal substances and also circulating ones. So the circulating ones are the humoral, and locally you can have, as we said, the paracrine effects. Remember, we said that you have gap junctions, especially in the circular muscle, so that communication from one cell to the next is quite easy. But the gap junctions are not as prominent, they're not that many in the longitudinal muscle layer. And what these gap junctions, is they permit the movement of intracellular regulatory molecules. So remember, we said the circular layer, when it contracts, it narrows the lumen. The longitudinal muscle, of course, contracts or shortens the gut, but it can only shorten it so much. Yeah, we can't have all of the gut contracting into a 1 foot long thing. So what I'm saying is that the most prominent change is a change in the lumen size. Now, the hormones and the neurotransmitters are the dominating components, and they act directly and indirectly on the muscle cells. Okay. Now, the hormonal influence and the interplay with the enteric nervous system takes place after a meal and also in between the meal. And I think I remember telling you this last week, that the cocktail or the mixture of gut peptides that are going to be secreted will change from meal to meal. It will depend on the content of the meal. And that is why you'll find that even some meals will affect you differently, even your mood and your well-being and the way you look, your perception of things, because of the interplay and the stimulation of the peptides that are going to be released. So people have realized as early as this is what is documented from the 3rd century. That's around which century is that? 300 AD, yeah. So, and they said that as houses which are well stored with provisions are most likely to be full of mice, so the bodies of those that eat much are full of disease. So that's why I was telling you, it is important that every once in a while you can go without meals or nibbling for a long period of time to allow the gut to deal with whatever's already in the lumen. Because if you don't, you just keep piling up, and the digestive system is not able to evacuate what you're taking in as quickly, what is in the gut as quickly as what you're bringing in or ingesting from the mouth. And they have realized that this can be the cause of many of the diseases of the gut. Now, the hormones or the peptides of the digestive tract are biologically active polypeptides, and they're secreted by nerve and gland cells in the mucosa. They act in a paracrine fashion, but they also enter the circulation, in which case then they'll have a humoral effect. Fortunately, they've got short half-lives. Approximately 5 minutes or so. So because they have that means that after 5 minutes, they will break down and they'll no longer be active. So this means that even if you eat a huge meal, all of those contents, they end up in the bloodstream, they'll not circulate and end up affecting the activity of your brain. So that's why I was saying, fortunately, they've got short half-lives. Now, unlike other chemicals, when they're given in large doses, they tend to have their actions overlap. So for example, some of them in small doses, some of these peptides can decrease motility. Others in small doses can increase secretion. But however, when you give them, each of them in large doses, you find that they affect more than the secretion. They affect more than the motility. So that's why I was saying when they're given in large doses, their actions tend to overlap. So sometimes it's difficult to know whether this increase in secretion was because of chemical A, B, and C, or what it was. So that's what has caused the problem in studying and understanding how they function. Now, typically the secretory cells, they're going to be found in the gut wall. They're typically flask-shaped, which means they're narrowest, the narrow end, the apical end, the sharp end is the one which is pointing into the lumen. So they've got a broad base. The base is what is going to sit on the basement membrane, and the narrow apex is one that reaches the mucosal surface. You have microvilli projecting from the apex into the lumen, and they contain receptors. So that's how they're able to know when they should release which particular substances. So there are very many classes of hormones. In fact, I think they've reached now probably about over 80, almost 100. We don't expect you to know all of them, but at least the major ones, which I shall mention here, the rest I shall refer to you to the textbooks. But they've got three major groupings. One is the gastrin family, and I think all of this is historical, depending on how they were discovered and the importance that was attached to them at that time. So you've got the gastrin family, and for example, here you have a gut hormone known as gastrin, and another one known as cholecystokinin. I'll give you the full name later on in this lecture. Then you have the secretin family. So you have secretin, glucagon, glycentin, VIP, GIP. And then you have others which cannot be classified in the gastrin family or the secretin family. And substances like these, are motilin, substance P, GRP, and guanylin. Now, the cells that produce these substances or these peptides are called enteroendocrine cells. And as we said, they're lining the whole gut. So in the stomach, you'll find them, in the small intestine, in the mucosa. However, whenever you find them in a particular region, they only produce one cell. So for example, the G cells are located in the stomach and they produce gastrin, and gastrin only. S cells are found lower down in the duodenum, and they secrete secretin. So you don't find one enteroendocrine cell secreting various types of peptides. So they only secrete one particular kind. Now, you have different forms of the hormone. Now, this is biochemically, yeah? And depending on the form of the particular hormone, it will have different effects. So for example, you can have the same hormone, but which will have, you can get one hormone which it's very similar apart from the molecular length. But that difference in length will also alter the actions of that particular hormone. Also, differences in the terminal ends will also alter the activity of that hormone. So you can have some hormones that are very similar, apart from that one has got a longer chain made up of more amino acids than the other. So that's how you're going to get differences occurring. So let's start by just looking at gastrin. As we said, gastrin is produced by the G cells, which are found in the mucosa of the stomach antrum, of the gastric antrum. Now, G cells, when they secrete gastrin then is the one which is responsible for causing secretion of hydrochloric acid. Now, the presence of hydrochloric acid in the stomach, in the lumen, will have a negative feedback on the G cells. So which means that when you have too much acid, or let's say the levels of acid, hydrochloric acid rise in the stomach lumen, that is going to inhibit the G cells and prevent them from secreting gastrin. So remember, the gastrin is the one which is going to be responsible for the release of hydrochloric acid. And remember, the gastrin, when it is secreted, it is secreted and it goes into the capillaries in the gut wall. So it acts as a hormone. It is not secreted so that it mixes with the luminal content. No. So what are the main actions of gastrin? One is the stimulation of pepsin and gastric acid secretion. So it is responsible for the initiation of protein digestion, chemical protein digestion. Gastrin also is responsible for mucosal growth in the stomach and in the intestines. So it has got a trophic action on the mucosa. The third thing is that gastrin does influence gastric motility. And in fact, when it is released, it actually relaxes the pyloric sphincter. So it enhances the movement of contents from the stomach into the duodenum. Then you have the final one, which is that the gastrin, once it's released from the G cells, it is also involved in contraction of the smooth muscle, not only in the stomach, but also in the stomach and the large intestine. So this, for one, gives you an example when I was telling you that the actions of these peptides is not strictly, it's not motility only or secretion only. Okay, depending on the concentrations, it can affect or can influence both secretion and motility. So this is what I was telling you about. When we're learning and we're writing down, we write secretion and we write motility as if they act independent of each other. No, they actually interacted them very closely. So it's difficult sometimes to separate one from the other. So gastrin affects motility, it affects secretion, especially of gastric acid, and also affects mucosal growth. Gastrin secretion is affected by the nature of the stomach contents. So if the luminal contents have a very high protein content, that is going to stimulate the release of more gastrin. If you stimulate the vagus nerve will also cause an increase in the secretion of gastrin. Okay, so those are two ways. One, the stomach contents can trigger the release of gastrin. Two, if you stimulate the discharge of the vagus nerve or stimulate the nerve, the vagus nerve. So as the protein is being broken down, receptors in the stomach will take in that information via the vagus nerve, which will then also cause an increase in gastrin release. And then the third way in which gastrin works is that you have blood-borne factors. For example, other hormones and calcium ions themselves can also trigger the release of gastrin. So those of you who've ever suffered from hyperacidity, usually people say drink milk because they think that is going to reduce the acidity. But because milk is going to contains calcium, you get it might cool off the burning sensation from the acid, but then later you get a rebound effect and you'll get more pain coming because the calcium ions themselves will cause stimulation of gastrin. So you can see that in order to regulate, or if you want to influence the release of gastrin, it's not just one way, there's three pathways. Okay, we said this content of the stomach. So if you want to release or to reduce gastrin release from the stomach, eat less protein. Or food, your meal should have less protein, because the more protein there is, the more it's going to stimulate gastrin secretion. Stimulation of the vagus nerve. So those people who are very anxious and stressed, that can also influence the release of gastrin. And then you have the blood-borne factors which are there. So you can see that it's difficult to, even if you do take care of one pathway, there's still two other pathways that can stimulate the release of gastrin. Okay, the next hormone is cholecystokinin, CCK. Okay, so when you see cholecystokinin, okay, the cysto refers to the gallbladder, chole refers to the bile. So cholecystokinin, you just always associate it with contraction of the gallbladder, but it does many other things. It's produced by the eye cells of the upper intestines and the nerves in the distal ileum and the colon. Okay, so not only in the upper intestine, it's because, or the upper half is because you've got the eye cells in the mucosa, but in the distal ileum and also in the colon, you have nerves which can release CCK. What are the effects? It causes contraction of the gallbladder and thus releases bile into the lumen. So remember, bile is produced by the liver. It is stored in the gallbladder. And while it is in the gallbladder, water is absorbed, so it ends up being concentrated, so that by the time the gallbladder contracts to release the bile into the lumen of the gut, it's actually releasing small amounts of highly concentrated bile. In addition to contraction of the gallbladder, CCK also influences the secretion of pancreatic juices. Now, pancreatic secretions are mainly of two types. You've got the one which is enzyme rich, okay? This is now contains all the enzymes which are going to break down the proteins, the lipids, the carbohydrates. And it's usually aqueous in nature, okay? But then you also have bicarbonate also being secreted. So some secretions will be alkaline and they produce the bicarbonate, and the bicarbonate is the one that is going to neutralize the acid that comes from, because the acidic chyme, when it leaves the stomach, it is highly acidic. So CCK increases the secretion of enzyme-rich pancreatic juice. At the same time, it also augments the secretion of bicarbonate-rich alkaline pancreatic juice. So it's going to produce from the pancreas both alkaline pancreatic juice to neutralize the acid and a juice that is rich in enzymes so that digestion can start taking place, chemical digestion can start taking place. Now, while this is going on, CCK at the same time will be inhibiting gastric emptying. So while it is enhancing and influencing the activity in the duodenum and in the early part of the jejunum, it inhibits gastric emptying so that no more chyme is released from the stomach until this amount that's in the duodenum has been processed. However, CCK is also found in the circulating blood. And what happens is that when the CCK gets to the brain, it influences the regulation of food intake, anxiety, and analgesia. So CCK is one of the examples I told you of peptides which are found not only in the gut, but also in the other parts of the body and where they produce different effects. And you can see in the brain, the effects are totally different from what is happening in the gut. Now, secretin is the next one, the next peptide. And secretin was actually the first of the peptides from the gut that was demonstrated to act as a hormone. And this was in 1902. So it was one of the very, very earliest, and people were very excited. And secretin is secreted by S cells, which are located in the glands of the upper small intestine. And secretion of secretin is also stimulated by products of protein digestion and acid. So if you eat food that is acidic, that has a lot of protein, once it comes into contact with the mucosa and the receptors are able to detect that, then you get the increase in secretion of secretin. What are the effects of secretin? Okay, it increases bicarbonate secretion. Okay, so which means it augments, it helps, it assists the work of the CCK, as you can see. Yeah, so it increases bicarbonate secretion in the pancreas and also in the bile tract, in the biliary tract. And this is what is going to play a role in reducing the acidity of the duodenal content. Because remember, if the contents remain acid for too long in the duodenum, the enzymes, when they come there, they'll be inactivated by the acidity. So that's why it is important that you neutralize that acidity from the stomach. And remember, HCl is coming from the stomach. So you neutralize the acidity so that the enzymes from the pancreas will be able to perform their duty. Now, that is also the reason why chyme is released into the duodenum in small spurts. We're talking about a few milliliters being secreted. So it's not being poured, it's being spurted in so that the bicarbonate can have enough time to neutralize the acid so that the enzymes will now be able to perform their functions. Secretin also decreases gastric acid secretion, okay? So, and it inhibits gastric emptying. So you can see those two help to prevent more emptying of the stomach. So the stomach holds things in until whatever's in the duodenum and the early part of the jejunum has been neutralized and a chemical digestion has taken place. Now, GIP is another interesting peptide. It's known, or it used to be known as gastric inhibitory peptide, because what they noticed is that when it was added or injected into the bloodstream, it caused an inhibition of gastric function. But now with more the experimentation and the technology that we have now, they renamed it so that now it is known as glucose-dependent insulinotropic polypeptide. Okay, now let's just look and see what it does. So it is produced by K cells, and it is produced in the duodenum and in the jejunum. So in response to food or the chyme being released from the stomach, it is secreted in response to glucose and fat. So if you eat a diet that is rich in glucose and rich in fat, when it gets to the duodenum, that content is going to trigger the release of GIP. In large doses, GIP inhibits gastric secretion and inhibits gastric motility. And it also stimulates insulin secretion. Now, this is interesting because you would think that you'd eat your carbohydrate, it is digested to its breakdown products, to glucose. The glucose is absorbed, it goes into the bloodstream, and once in the bloodstream is when you're going to get now the trigger for insulin to be released. Yeah, the pancreas will be stimulated and insulin will be released to bring the sugar levels down. Now that is like living dangerously, living on the edge, because you need to control the blood sugar levels very, very strictly. So what this suggests is that even as you're digesting your food, when the food gets to the duodenum and jejunum, the body is able to, you have receptors, so you can get information regarding the amount of carbohydrate or glucose in the food or in the luminal content. And if it is too high, while the glucose is still in the gut lumen, messages will be sent to the pancreas to start releasing insulin in the blood so that the glucose, when it is absorbed, you don't get those high spikes, okay? So what we're saying is that there's crosstalk between the gut lumen or the contents in the lumen and the pancreas, which is quite far removed from the lumen. And so it also suggests that your body can prepare. So when you're chewing and eating, this information comes right from the mouth. If you chew and you swallow, your receptors in the taste buds will be sending information to the brain so that by the time this food now is being released from the stomach into the duodenum, the pancreas has also been involved, not only to produce digestive enzymes, but the endocrine pancreas will be asked to secrete insulin. The next one I'm going to talk about today is next peptide is vasoactive intestinal peptide. And as you can see, vasoactive intestinal polypeptide, which means it acts on the blood vessels. Now, it is found in some of the nerves of the digestive tract, and also it is found in some of the neurons in the brain, those, especially those that contain acetylcholine. Okay, so in the brain and in the autonomic nervous system. But in the digestive system, you don't find VIP coexisting with acetylcholine in the nerves. Okay, so what we're saying is that where you find acetylcholine and VIP is in the brain and in the autonomic nerves, but not in the digestive system. Now, vasoactive intestinal polypeptide is released when fat or the fat content is high in the duodenum. And the effects of this is once the fat stimulates the release of VIP, you get intestinal secretions being stimulated. And these intestinal secretions will be rich in electrolytes and water. You also get relaxation of the smooth muscle and the sphincters in the small intestine. Another effect is that VIP dilates the peripheral blood vessels and it inhibits secretion of gastric acid. Okay, so you can see even as we're going further away from the stomach, you're still getting information from the luminal contents, which can go back via the enteric nervous system to the stomach to inhibit secretion of gastric acid. So you're seeing here, there's still a lot of crosstalk, a lot of information being passed on from region to region, even when you have distance between the different parts of the gut. Another one I'm going to touch on is motilin, another gut peptide. Now, motilin is secreted by the enterochromaffin cells in the stomach and also by the MO cells in the stomach, the small intestine and the colon. Okay. Now they say that these are the same ones that would secrete serotonin. Motilin acts on G protein coupled receptors on the enteric nerves of the duodenum and the colon. Now, once it is injected into the animal or the person, the subject of observation, motilin produces contraction of both gastric and intestinal smooth muscle. Now, because it causes contraction, okay, so motilin has been associated with motility. So motilin, motility. It has also been thought to be the one that is responsible for the stimulation of the mean migratory complex. Remember the motor complex or the mean migratory complex or the motor, it's got so many names, migrating motor complex. The one which we call the housekeeper of the stomach. The one which we said starts after you finished eating the meal. Between that time and the next time that you eat, you have a ring of contraction that starts from the stomach and goes all the way down to the colon to propel left remnants or to help clear the lumen of the gut in preparation for the new meal. Now, erythromycin is a drug that is used and we usually prescribe it, but one of the side effects or one of the effects of erythromycin is it stimulates motilin receptors, so that if you have people who have got decreased gastrointestinal motility, if you give them erythromycin, it will bind onto those motilin receptors, stimulate the release of motilin, and as a result, increase motility along the gut. So, or else we can say that one of the side effects of taking erythromycin is an increase in gastric motility. And you can see why. It's because it is able to bind onto the motilin receptors. Now, I think that takes care of that. So I have only summarized the ones which I thought were key and prominent. But when you go back to your textbooks, you'll find them many. If you go onto the internet, there's a whole range there. Every day they're discovering new ones with names that sometimes we can't even remember. In the beginning, we also told you that these neuropeptides, or let's say these gut peptides, can be either stimulatory or inhibitory. They're stimulatory on the intestine, okay, in the contraction, or they can inhibit. So they can enhance the motility or they can decrease it. I guess they can also do the same when it comes to secretion. So this particular slide just gives you an example of some of the neuropeptides, the ones on the left column. And you can see that some of them, they're substances that you've come across in other parts of the body. But this list gives you the ones that are purely stimulatory when it comes to intestinal contraction. So including histamine, motilin, opioids, serotonin, acetylcholine, okay? And you can look at the list. Then similarly, we've got another list of those that are inhibitory in nature. For example, you've got your calcitonin gene regulated peptide, you've got your GABA, glucagon, okay, nitrous oxide. So the whole list, secretin, somatostatin, VIP. So you can see that when in one meal, you're actually producing a cocktail of this mixture of both inhibitory and stimulatory, but depending on the meal, the resultant concoction can either be inhibitory or stimulatory. So I think that is the end of this. For those of you who might be interested in seeing the role of these substances, they play a role in appetite, gastric motility, and many other things that affect more than just the digestive system. Okay, so these, you can also look up these two papers. Any questions so far? Yeah, I'd like to do as much as possible. So if we have done these ones, yeah, we can take a, I'll try and go a little more slowly now, okay? Because remember, we're looking at motility and secretion as if they're two different things, as if they're two separate functions of the gut. And yet they're so closely related that it's difficult, it's difficult to study one without affecting the other, okay? So let's move on now and talk about the hepatic portal circulation, and also just one or two things about the gut microbiome, which is what people are really interested. This is the interest right now, and they've realized that the gut microbiome affects the human being so much more than what they thought. So most of this is you need to go and revise your anatomy. And if you remember, the liver has got two sources of blood. You've got blood coming from the gut, carrying all the digestive end products, and this comes to the liver via the hepatic portal vein. You also have oxygenated blood, now coming from, as the blood comes from the lungs, and it travels now via the hepatic artery, and this is what is going to supply nutrients and oxygen to the hepatocytes. And then the liver is drained by the hepatic vein, okay? So you've got two sources of blood from the hepatic portal vein and from the hepatic artery, and then it's drained via the hepatic vein. Eventually it ends up in the vena cava, and that's how it ends up on the venous side of the circulation. So that is what is important. And when you've had a heavy meal, when you've eaten your stomach, and your stomach is full, then the hepatic portal vein, the blood flow from the hepatic portal vein is going to increase. So knowing that, that circulation, which I showed you just now in that illustration, is known as the splanchnic circulation. So it is the blood flow, or it's part of the splanchnic circulation. It's the blood flow to the gastrointestinal tract, to the liver, to the spleen, and also to the pancreas. Okay, because they're all going to be involved in digestion. Now you have two large capillary beds, which are partly in series with one another. The small splanchnic arterial branches supply capillaries to those organs. And from these capillary beds, the venous blood flows back to the portal vein and produce most of the blood to the liver. So the blood that is coming into the liver, majority of it is coming via the hepatic portal vein. This is also important for you because if you have blockage in the hepatic portal vein, then you can end up with venous hypertension, which is also not good. So the hepatic artery, as I said earlier, supplies oxygenated blood to the hepatocytes. Now, I always believe that a picture probably says more than 1,000 words. And this is a resin cast that was made of an arteriole bringing blood to the gut wall. So obviously this is now via the mesentery, and you can see the blood vessels here. You got different calibers, but I think this is the level of the arteriole. And once the blood vessels go into the wall of the gut, they then branch out into smaller caliber vessels until they form the capillary network. So you can see this capillary network now. Now, can you imagine every time when you're eating, the blood has to flow through here. And this is why I was telling you that it's important for the tonic contractions, because they're going to help propel or to get movement of the blood through this part, even as the blood vessels, as the gut is undergoing its function. So you can see now that if you have a blockage, for example, if this is the blood vessel that is going to carry blood away from this portion of the gut, going towards the hepatic portal vein, if you were to block it, you would get backlogging of blood, you get hypertension. Not hypertension. Hypertension is more central. What you'd get here is a blockage, and these vessels would all become engorged. And since these are capillaries, you'd end up with filtration and fluid leaving the blood vessels and pouring into the peritoneal cavity, and you can end up with ascites. So I just wanted you to appreciate that. So when we're talking about blood flow, you know, it's not just a series, it's not just two pipes which are traveling along. You can see the rich network of blood vessels, of capillaries in the vein, in the wall of the gut. So imagine the entire length of the gut followed or comprised of such vessels. Now we have neuroregulation of blood flow, okay? And the control of the mesenteric circulation is exclusively through the sympathetic stimulation. And remember, when you stimulate sympathetic nerves, what happens? You get vasoconstriction, okay? So you're going to constrict the mesenteric arterioles and the capacitance vessels. Now, this response is mediated by the alpha receptors and beta receptors, but primarily the alpha receptors are the ones that are more responsible. So when you stimulate the sympathetic nerves, what you're going to get is vasoconstriction, okay? You narrow the lumen of the blood vessels. Now, you can also have autoregulation taking place, but the autoregulation is not as well developed as what is found in the brain or even in the kidney. Because I don't know, I think you've done renal. Yes, I think you've done renal physiology. And you know that autoregulation there occurs very, very efficiently. So how does autoregulation occur in the gut? It is primarily via the metabolic principle, which is responsible for autoregulation. So what is the metabolic principle? I'll refer you also to the books to go and read more about this. But basically what happens is that as you get more and more metabolic byproducts, for example, as the meal is being digested and you're getting breakdown of more, so you're getting a buildup of amino acids and lipids and glucose and fructose. All of those, as they are building up in the bloodstream, you're getting an increase in these metabolic byproducts. They're going to be responsible for vasodilation of the vessel so that they can be moved or ferried away from that particular region. So what I wanted you to realize or to take home is that vasoconstriction is as a result of sympathetic stimulation, but relaxation and vasodilatation of the vessels will occur as you get a buildup of metabolic byproducts. Again, this picture has come again. I think we discussed this last week, but I'd like you to go and study it again just to get a picture of, I think once you understand the anatomy and the microscopic anatomy, then it gives you a better understanding of what is happening in the gut, okay? Now, there is something that is known as functional hyperemia. And what happens is that, for example, if I just loosely try and explain this, if you were to open a window into the stomach and you could see from outside, if this was the interdigestive period, when you looked at the mucosa, it would appear pale. But the minute that person starts eating or drinking a fluid, whether it's a smoothie, whether it's a soda or water, of course, water doesn't have any nutrients, but anything that requires digestion, what you'll notice is that the mucosa of the stomach will, or even of the gut, as the contents arrive there, will become pinker and redder as the intestinal blood flow increases. So what we're saying is that when you have the lumen acquiring, or there's more luminal content coming into a particular area, apart from the motility that is going to take place, you're going to get an increase in the blood flow. Because remember, the blood flow is going to be necessary because you're going to have an increase in secretions, an increase in motility, and also eventually you're going to have an increase in absorption. So the blood flow to that particular part of the gut will increase upon demand. And initially when they were, thinking, because people used to think that because the gut has got this muscular coat, the outer longitudinal and the inner circular, that when these muscles are active, that is when the blood flow is going to be most. And also that if you were to supply blood to the muscle, to the gut, most of that blood is going to go to the smooth muscle because the smooth muscle is the one which is active. However, on experimentation, they were quite shocked because what they found is that 2/3 of the blood that goes to the gut actually goes to the mucosa, not to the muscle layer. Only 1/3 goes to the muscle layer. And that's why I was telling you about this term that is known as functional hyperemia. So intestinal blood flow increases on ingestion of food in order to facilitate smooth muscle contraction. Ingestion of food also stimulates the secretion of gastrin and cholecystokinin. These cells that are going to secrete these gut peptides are found in the mucosa. You also get absorption of food, which while the food is being absorbed, it's going to be absorbed together with water. And this is the breakdown products of food. So you get an increase in intestinal blood flow. And that is what gives functional hyperemia. And it is mediated principally by glucose and fatty acids. And this is what is known as the metabolic principle. Because remember, we said this is what is going to cause vasodilation and to facilitate or to allow more blood to come into the area. This is just another picture similar to what I'd shown you earlier to show you the arterioles coming and breaking down into smaller caliber vessels until you have this rich capillary network all in the gut wall. Okay. Now, if we look at a picture of a villus, okay, so one villus, many villi. The villus is a finger-like projection, as we said, and the villus is lined by epithelial cells, which will have microvilli on the side that projects into the lumen. And that's how you're going to get an increase in the absorptive surface or into the surface that is going to interact with the food. Now, once substances are absorbed, they're going to go into the center of the villus and they will be directed into the interior of the body and be ferried away from the gut. So the villus has got blood vessels in it. You have the arterial, which will give off branches, and the arterial is the one carrying oxygenated blood, okay, which is the one in red. You'll also have the venule, which is draining the villus, okay, and will end up draining into the venous blood flow of the body. And in between, you will have a branch of the lymphatics, which is known as the lacteal. Okay, so basically what happens is that the products of digestion of carbohydrate and protein will be directed into the venous side. Okay, the arterial branch is going to be bringing oxygen and nutrients to this part of the body. Now, the byproducts of fat, when fat is broken down, the fat byproducts are not going to be directed into the veins because you'll end up with too much, okay, I don't know whether it's because, but you'll end up with too much fat in the bloodstream. So the fat byproducts are going to be directed into the lacteals, and the lacteals will join up and become part of the lymphatic system, and the lymph will finally drain in the thoracic cavity into the venous blood. So that is how the blood, or how the body tries to reduce the amount of fat that is going to be found in the blood. Now, what is interesting about this villi is that there are certain lengths, as you can see. Now, when the nutrients and the oxygen are coming through, you expect them to come in from the arterial end to go all the way to the tip, and then come back down, now the byproducts to be, metabolic byproducts to be carried by the veins and all the way back. But just like human beings like taking shortcuts, some of these molecules also like taking shortcuts. So they'll probably come in and maybe only go back of the way and go down. So what we're saying is that as you go up to the tip of the villus, you'll have a graduation, an oxygen gradient, so that the cells at the tip of the villi will always be operating in an environment that has got less oxygen than those which are close to the base. Now they're specialized, they're adapted to that, and that is how they function. And so long as you're healthy, life will go on, life will be good. But if for some reason or another you get a compromised blood flow and the oxygen levels get less than normal, then the cells at the tip of the villus are the ones which are going to be affected. They will become necrotic, apoptotic, and what will happen is that you'll have a shortening of the villus. And as you can see from this picture, if this villus gets shorter, let's say even up to this line here where it says lacteal, that is a large number of epithelial cells that have disappeared. And remember, each of these will have microvilli. So the absorptive surface area will be diminished drastically. And if this happens in a large portion of, let's say, the small intestine, you can end up with someone who's eating a perfectly ideal diet, balanced in quantity, in distribution, and everything. But because the absorptive layer in the small intestine has been compromised, they're not able to absorb enough nutrients. So they can still end up with malabsorption if this is what is happening in the gut. So that's where you have now problems with the irritable bowel syndrome, the Crohn's disease, because what it does is it affects the villi, it affects the mucosa. And even if you're giving them a perfect diet, they can still end up with diminished absorption. So this, I think, explains that, yeah? So you have countercurrent flow in the villi, blood flow, and the vessels are in close proximity. So at the base, the oxygen easily diffuses from arterial into venule without necessarily traveling to the very tip of the villus. So in disease conditions, an oxygen deficit easily results in ischemic death of the cells of the villi, and you end up with short, blunted villi. This is what happens, for example, in Crohn's disease. So the net result, as we said, is poor absorption of luminal contents, despite the luminal contents containing all the nutrients that may be required by the individual. So they could be eating a perfectly good diet, but still you wonder why is their body not responding, or why are they still getting disease? Why are they still showing symptoms of absorption, of malabsorption? So this is just to show you what the villi look like when they're many. So you can imagine if all of these are shortened, these are very, very many villi in this very short space of time. So this is just a tiny cross section of the gut wall, and to show you how if you have diminished blood flow, or you get a shortening of the villi, the enterocytes die off, you actually are going to cause a severe nutritional deficiency in that person. And this is now just a histological picture showing you the same thing, okay? So this is the center of the villi, so you can see the enterocytes lining the lining. You can see deep in the submucosa here is where you're going to find your glands, which are going to empty their secretions into the gut, into the lumen. So their secretions are going to mix with the luminal contents. And this is the beginning of the muscle coats, okay, the inner circular and the outer longitudinal layer. No, actually, this is not. This is actually the muscularis mucosa, which is also arranged in two so that you have an inner circular layer and an outer longitudinal layer. Okay, the greater muscle coats are deeper, way below the bottom of this slide. Okay, so now let's look at the liver per se. Okay, so we move from the intestine. Now we're in the liver and how the liver functions. So if we look at this diagram and remind ourselves of the anatomy, the hepatocytes are these cells here, the clear, was it beige colored epithelial cells here, okay? And you have the blood vessels. So you've got the blood coming in. Remember, the hepatic portal vein is going to be carrying the digestive end products. You have the hepatic artery, branches of it, bringing in the oxygenated blood. And once these vessels come into the tissue of the liver, the blood now pours into sinusoids. It's no longer contained in vessels. It actually pours into sinusoids. And by pouring into these sinusoids, it means that it's able to flow a little more slowly and also have now more intimate interaction with the hepatocytes, which are lining the sinusoids. Okay, so the blood is going to be coming from here. You can see the hepatic portal vein, a branch of it, and a branch of the hepatic artery. The blood will pour and then travel to the center of the functional unit, which is the acinus, and the blood will now eventually drain into the central vein after having been processed by the hepatocytes within the sinusoids. Now, as the hepatocytes perform their functions, the myriads of function, detoxification, etc., they also produce bile. And the bile, as it is formed, will travel in the opposite direction to the flow of blood. It will be traveling from the center of the acinus to the periphery, where it's going to join branches of the bile duct. And then those branches will join eventually and leave the liver as the bile duct. Now, what you need to remember is that the liver produces bile 24/7. It is continuously producing bile. However, the bile is going to be dilute, and once it goes from the bile duct and directed to the gallbladder, not the bile duct, it's going to be directed to the gallbladder, it will be stored there. And as we said, water is absorbed, it will become thicker and more concentrated, so that when you have contraction of the gallbladder, once you have a fatty meal, it's only a small amount of bile that is going to be required to emulsify the fat in the meal. So you've got to have this countercurrent flow of blood flowing from the periphery of the acinus to the center of the acinus, and the bile, as it is forming, traveling in the opposite direction. Now, if we look at this other picture here, this is now a diagrammatic form, and it shows you the functional unit of which is known as the hepatic acinus. And you can see in the center of each acinus is a branch of the hepatic vein. Okay, so all these ones in the center will be branches of the hepatic vein. And on the periphery of the hepatic acinus is where you'll have a branch of the hepatic artery carrying oxygenated blood and a branch of the hepatic portal vein. So that blood will be traveling centrally, okay, from the periphery to the center of the acinus. But the bile, which is the one in green, will be traveling in the opposite direction. It will be traveling from the center of the acinus to the periphery. And that is how physiologically, that's how the hepatic acinus functions. So I'll leave you to go and refresh your anatomy so that now you can appreciate the functions of the liver. Okay. Now we have here regulation of blood flow. So what we're saying is that the portal venous system does not auto-regulate, but the hepatic arterial system does. Okay, now I'll leave you to go and read more about that. But what I wanted to know, what you needed to note, is that the liver, because of the tissue, then it's a friable tissue, actually contains 15% of the total blood volume of the body. And in humans, the liver is considered as an important blood reservoir. About half of the hepatic blood volume can be rapidly expelled in response to hemorrhagic loss of blood. For example, if someone breaks their femur or they're involved, they can, if they're losing a lot of blood, the liver can contract and release at least half of that blood, which can then be, can help maintain the blood volume. So in other animals, the spleen does the same job. Hello. Just a minute. Just hold on, you guys. Okay, just give me, okay, we had wanted a break, so can we take it from now? Is okay, level 2?
If we took it now, would we come back at 4:15?
Just give me a minute. Let me find out, because this is the department that's calling me. Just hold on. Slides, about maybe 2 or 3. And then we can take the break until 4:15, okay?
Okay.
Wow. Okay, so the slide, I think we're still there. We're okay, okay. So what we're saying is that you've got unusual circulation in the blood vessels, because most of the blood, instead of returning directly to the heart, it is directed to the liver via the hepatic portal vein. That's what makes blood from the gut different from blood from the other parts of the body, okay? So that's the uniqueness of the splanchnic circulation. Now, during fasting, the liver receives about 1.3 liters of blood per minute from the portal vein, and only 500 mLs from the hepatic artery. So majority of the blood is actually coming from the digestive system. So the more you eat, the more you ingest, the more you're loading your hepatic port or overworking your hepatic portal vein. This just tells you about the liver, the pressure differences. And remember, we said once the blood goes into the liver tissue, because it's no longer contained in the vessels, then the pressure, there's a drop in pressure, because what you have in the liver is sinusoids, not blood vessels per se. If you have cirrhosis of the liver, when you have damage to the liver, then the cells are replaced by fibrous tissue. So you get a decrease in the hepatocytes. And also the fibrous tissue doesn't allow the blood vessels to dilate as they should. And so you get problems. So cirrhosis of the liver can result from alcoholism, chronic alcoholism, ingestion of poisons, for example, carbon tetrachloride, viral diseases, for example, infectious hepatitis, and also in obstruction or infection of bile ducts. So all of this is going to destroy the cells. And remember, the hepatocytes are actually epithelial cells, yeah? And once you get damage and you get the liver not being able to dilate and accommodate this blood, you can end up with hot portal hypertension. So you get the blockage of the portal system, either by a large clot in the portal vein or any of its branches. And if this is sudden, it can lead to impedance of blood from the intestine and the spleen. The capillary pressure in the intestines is increased, as we said, from the backlogging. And the result is extravasation. We said fluid leaves the blood vessels. And because of the buildup in pressure, you get so much fluid loss it pours out into the, you're going to get the fluid accumulating in the peritoneum. You're going to get ascites. And the patient can die within a few hours due to excessive loss of fluid from the capillaries in the lumen, which is the reason why I showed you that picture of the capillary network in the wall of the blood vessels. And remember, capillaries are leaky, so they can contain the proteins and the other larger macromolecules, but the fluid will just flow through. So you can end up with ascites, and they end up with hypovolemia and many other complications. So portal hypertension is not a nice thing. And just for you who like pictures, this is actually an example of a patient with a severe case of ascites. Okay, other facts about the liver. It's capable of restoration. So they say that if you lose 50% of the liver, it can replenish itself back to normal size given time, and if there's not continued trauma. And 50% of all of the lymph that is formed in the body occurs under resting 50% of all the lymph that is formed in the body while under resting conditions is derived from the liver. And the liver is capable of remarkable restoration, as we said, so long as the damage is not complicated by viral infection or inflammation. Okay. And they say when 70% of the liver, is resected, the organ has been shown to repair and restore itself to its normal original size. So it has a potential for repair and regeneration that is greater than other parts of the body. Then you have the large phagocytic macrophages, which line the hepatic venous sinuses. So even as the hepatocytes are performing their functions, these macrophages are cleansing the blood of bacteria. And what would be the source of this bacteria? Mostly it's from the colon, the microbes in the colon. Okay, and then finally, just a small note about the gut microbiome, which basically represents, this is all the resident microbes along the digestive tract. So we have germs, we've got microbes in the mouth, in the pharynx, in the esophagus, in the stomach, in all along the gut. And the population that is in each region differs. Okay, so you don't have the same, you won't have the same microbes, for example, in the esophagus as you would find in the colon. Okay, but you've got symbiotic relationship that exists between you, the host, and the microbes in the gut. So initially they used to think, oh, you've got microbes in the colon. The colon is the dirtiest part of the gut, but they've realized, no, that actually those microbes are actually quite useful. They play a role in completing the digestion of what you were unable to digest. If the luminal contents get into the colon and digestion has not been completed, the gut microbiome in the colon will take care of that. While they're digesting, doing the final digestion and digesting things like cellulose, which we cannot digest, they also produce short chain fatty acids, which will be taken up and used by some of the colonocytes. And they also produce vitamins, which are then absorbed in the colon and will end up in your bloodstream and help you. So that's why we're saying we've got a commensal a symbiotic relationship, a win-win situation where the host helps the microbes and the microbes helps the host. Now, the species and the number vary and differ along the gut. And for example, the ileocecal valve is very important because it keeps the colonic microbes out of the ileum. And remember, this is where the small intestine joins with the large intestine. And remember, we said the colon has got a much larger diameter than the small intestine. So imagine if there was pressure buildup in the colon, it would be very easy to get backlogging of the luminal contents coming backwards. And that would be dangerous because of the microbes that would then invade the ileum. Remember also, at the junction of the ileum and the and the colon is where you find the appendix. And the appendix is kind of like a blind loop. So every time you eat a meal, a sample of your luminal content goes into the appendix, which is filled with lymphoid tissue, which can then sample the contents of the lumen. And in that way, the immune system keeps up with what is happening with the luminal content along the gut. Now, what is interesting is that when it comes to the number of microbes, for every human cell, there will be 10 microbes in the gut alone, which means that if the cells that contain your DNA in the gut will be 1/10 compared with the cells that contain what's it called, genes from microbes. So basically what we're saying is that your microbial population in the gut is greater than the cells of your body. So the genetic load is much greater than the individual one. And this is very interesting. So you start wondering, if in the gut, if you count all the cells that make up your body from your hair all the way to your toe, and then compare those with the gut microbes, the number of gut microbes is 10 times greater than the cells that make up you. Now, if you were to count now the microbes in the genitourinary tract, in the respiratory tract, the ones lining your gut, the ones lining your skin, then you can see that actually the cells that make up you as an individual are a fraction compared with the other microbes. We're like little tiny colonies. We're like a little universe. Each individual of us is like a universe with full of very many microbes. And so even when you're eating, you've got to remember, you've got to keep everything in balance, because when that balance falls out, that's when you get disease. So the gut microbes play a key role in immunity, and the colonic microbiota, as I said earlier, perform the final digestion. They synthesize nutrients, and they also secrete peptides, which are responsible to prompt your, or that keep your immune system healthy, and the nutrients will be absorbed and they will be used by your body later on. So I'm always telling people, when you're walking around feeling yourself and thinking, I am so and so, and your ego is really huge, remember, you're made up of more microbes than you are you. So it's just something that is interesting. And all these that are discovering now, that's why the gut microbiome is what everybody is concentrating on right now. And this picture, I'll leave you to have a look at later. It just shows you a healthy gut and a gut with an altered microbiome, altered environment. So in a normal gut, when you're just eating normal food, because remember we said food and water are not sterile, you have your physiological microbiota, and they keep the pathogenic microbiota in check. And you can see here, they actually even outnumber the pathogens, or the ones that would cause disease, which are the purple ones. And then this is your healthy epithelial barrier. And behind that is where you have now your part of your immune system, your plasma cells, your T cells. And so what we're saying is that every time you have a meal, because you're introducing different things and foreign things, what you produce during digestion, or what happens in your gut mucous membrane, is you have a physiological inflammation. Okay, so just like how we say blood clotting is a normal phenomena, but if the blood clots in your veins when you're working, that is not normal. It's the same thing. This inflammation is occurring all the time, because you're always exposing, every time you eat, you're exposing foreign substances to your immune system. So it is physiological inflammation, and it will happen with every meal, but you don't have any damage to your mucous membrane, and your body is otherwise healthy. However, when you have altered gut environment, now altered by what? By poor diet, for example, eating a lot of junk food, highly processed food with low nutrients or no nutrients, poor hygiene, eating food that is infected, spoiled, whatever. You've taken antibiotics, because when you take antibiotics, they will be absorbed and go into the bloodstream and work and deal with the invading microorganisms. But in the gut lumen, they will also have effects on the luminal gut population. So that, or even pollutants, viruses, infection, what you're going to end up when you alter the gut environment is what is known as a dysbiosis, where you have a decrease in the peacekeeping bacteria. And so this is now shows you the difference, the balance between the environment and the host immune system, and why it is that you can get disease. And you can see here the pathological inflammation, where you're getting damage even to the mucosa, which will affect even gut function. And yes, I think that is it. Okay, now I can't remember. These are some videos I found on the internet, and as you can see, I think I have the link somewhere, I can't remember. But if you just Google and put in, YouTube videos, food for thought, it's a video, and this is the speaker, Roairi Robertson. There's another one, Altering Microbes by Elaine Hsiao. I think most of these were TED Talks, so they're about 10 minutes, 15 minutes max. And then there's also another one by Rob Knight, who was the first person to experimentally show the importance of the gut microbiome and how it changes, for example, even from birth until the baby is weaned, and how long it takes for you to get an adult typical microbiome, and what happens when someone takes antibiotics, what happens to that microbiome population. So I would highly recommend that you key in these. Just Google and go to YouTube videos and see that. And if you have any problems, let me know, and I'll see whether I can actually locate the links that I had for these movies. So I think at that point, we're now at, yes, time. So we can take a short break, and then we can be back in, is it 15 minutes, class rep?
Yes.
Okay, so 15 minutes. Okay, that's good. So let's take a break. I'll also go and drink some water. Thank you. But what I wanted to do just now is so that tomorrow tomorrow will not be as heavy as today. So if we finish with the motility today, then tomorrow we'll just do about secretions, okay? So what we're going to do now with this series of slides is just to go through the gut, starting from the mouth, and just describe what kind of motility takes place, okay? So in the mouth, we have mastication, which is a reflex behavior. It's called semi-reflex because, hello?
Hello, sorry, are you sharing your screen?
I haven't shared the screen with you.
Oh, no, not yet.
Just hold on, okay. So are we looking at the screen right now? You can see it?
Yes.
Okay,
yes.
okay. This is what happens when you've had a long day. Okay, so we're talking about motility, and we're just going to go quickly through the regions of the gut and see what happens there, okay? So what we're saying is that in the mouth, the process that takes place is basically mechanical digestion in the form of mastication, which is another word for chewing. And mastication is a semi-reflex. Okay, they say reflex behavior, but it's a semi-reflex because you have control of initiating. You're the one who opens your mouth, puts food in, and then you start chewing. But once you start chewing, you're not consciously in control of, saying, press hard, this is meat, or this is jelly, don't press so hard. No, so that's why it's called a semi-reflex, okay? Now, the mouth, you've got skeletal muscles, the muscles of facial expression, the muscles of mastication, which are involved in not only crushing the food between the occlusal surfaces of the posterior teeth, but also the buccinator and the lips help prevent the food from falling out of the mouth, which is why I guess when we were growing up, you're always told don't chew with your mouth open. Not only because we don't want to see the contents of your mouth, but because when you chew like that, then food just splutters and falls out of the mouth. So when the food is chewed, it falls into the floor of the mouth, under the tongue, or it falls into the cheek region. The buccinator tightens, the tongue lifts the food up, or the floor of the mouth up, and the food is placed back on the posterior surfaces of the teeth, and you keep chewing until the food is fine enough for you to be able to form a bolus. So how do you know that the food is fine enough to form a bolus? usually you rub the food between the dorsum of the tongue and the hard palate. You have palatal rugae, and the tip of the tongue, or the tongue, is sensitive so that it can tell how fine the food is, whether the particles are fine enough for you to form a bolus and then be able to swallow. You also have proprioceptive receptors in the periodontal ligament and also in the muscles of mastication. And these are the ones that give you information as to when your teeth are touching, or when the jaws, when the teeth have broken through the food and the teeth have come into contact. So we should now relax so that the jaw can drop, and then the process of the cyclic process of mastication takes place. The main functions of mastication, as we said, was mechanical digestion, breaking the food into smaller particles. So you can actually start the mechanical digestion in the kitchen, on your plate with a knife and fork, and then in your mouth. And while you're chewing and breaking the food up, you're also mixing it with food, with saliva. The saliva acts to moisten the food, and also initiates chemical digestion, because in saliva you have ptyalin, which is also known as salivary amylase, breaks down carbohydrates, and initiates the breakdown of carbohydrates. You also have lingual lipase, which helps in the feel of the food. When food has a little oil in it, it's easy, it's palatable, it feels better in the mouth, which is the reason why when you present people with a plate of boiled food, they just look at it and they turn up. Yeah. But even when you steam food, if you just put a little sprinkling of little bit of oil, it makes it pleasant, not only to look at, but even to taste and the feel in the mouth. Because remember, we said you have a lot of receptors in the oral cavity, and all those receptors carry a lot of information to the central nervous system regarding the food, even while you're chewing it. So those of you who don't chew enough, you're shortchanging your brain. You're not providing enough enough material, enough information to your brain, and your eating becomes mechanical. In the main function of mastication is also the lubrication and moistening, the formation of the bolus. The saliva contains mucus, so that the food does not stick to the mucous membrane in the mouth, does not stick to the hard palate. And that's why you'll discover that if you're tense, if you're going into an exam and somebody gives you a biscuit to eat just before you walk in, you'll probably hate them, because when you chew the biscuit, there'll be no saliva, because your sympathetic nervous system is triggered. It's going full throttle. The saliva production is diminished, and you'll even find that talking is difficult, because your tongue tends to stick to the roof of the mouth. Okay, now the rhythmic jaw movements of mastication are controlled by the brainstem areas. And remember, we said we're talking about skeletal muscle, which is the reason why it is a reflex function. And you've been chewing for such a long time, from when you were born, that even now you don't think about it. And because you're not thinking about it, you're not conscious. If you're distracted, for example, when you're watching TV or listening to somebody telling a gripping story, sometimes you find you've bitten your tongue. Or if you're eating food that is you're in a hurry, or you're really hungry, you find that you bite yourself. Usually the cusp of the canine or the bicuspids, usually it's the corner of the mouth. And what is interesting is that once you bite yourself once, chances are the next time that you bite, you're going to bite the same place. And it's very painful, especially since you're biting yourself unaware. So that is what is known as lazy tongue syndrome, lazy lip syndrome. So you should always focus on your food when you're eating. Don't go with your mobile phone, stop watching movies and eating at the same time, because the flood of information going to the central nervous system becomes confused. For example, if you're enjoying a meal and you're watching a movie where people are killing each other, there's blood and guts all over the place, accident, loud noise, it will also affect your enteric nervous system. So you can, especially at night, and you go to bed and then you can't sleep very well, you can end up with nightmares. And it's all correlated. It's now that people are able to explain those things that are happening. After we chew, normally we swallow, okay? So deglutition. Deglutition is the technical term for swallowing. This is also a process that is reflexly coordinated by the swallowing centers in the brain. And why it is reflexly coordinated, okay, let me say this before we actually look at the slide, is because the pharynx is a common chamber for the respiratory system and the digestive system. So the air has got to come from the nose through the pharynx into the larynx and into the thorax. The food goes from the mouth through the pharynx and then goes into the esophagus, which is behind the trachea, and then goes now into the digestive system. So because you've got so many muscles, okay, they're all skeletal that are going to be involved, but you've got to rapidly contract them and relax them, because as you swallow, you're going to temporarily hold your breath as the epiglottis closes the entry into the trachea, and then the bolus rolls off the back or the top of the epiglottis, and is directed, over the closed trachea into the esophagus. The trachea, remember, has got cartilaginous rings, so it is patent. The esophagus is normally squashed. It's not usually open until the bolus is going to go in. And then once the bolus passes through the top of the esophagus, it collapses again. So in order to prevent food from going into the airway, you have to have reflex coordination so that the back of the nose, the food doesn't go retrogradely into the back of the nose, and the contents, the food content also doesn't go into the larynx, which is why it's covered by the esophagus. So for all of that to happen, it is reflexly coordinated. And fortunately for you, you don't have to think about it, so that you can have a smooth swallowing. And just to show you how coordinated this is, you should try laughing, or someone cracks a joke when you're drinking a soda, and then the soda goes down the wrong way. Yeah, it comes out either through the nose, and your nasal mucosa is not geared to handle carbonated drinks, so it's painful sometimes. It's because of the carbonic acid and the bubbles breaking up there. So that's what I'm saying. That's why it's important to have reflex coordination. You have 3 phases. These phases are determined by the location of the bolus at that particular time. So the bolus passes through the oral cavity, then through the pharyngeal cavity, and then through the esophageal conduit, which is why you have the 3 phases: oral, pharyngeal, and esophageal. Of these 3 phases, when the bolus is passing through the oral cavity, that is when you have control, voluntarily control over the bolus. You can change your mind. You can chew, decide, I'm not going to swallow this, and spit it out. You can chew, form the bolus goes to the junction between the oral cavity and the pharyngeal cavity, and then you change your mind, in which case then you gag and the food comes back into the mouth, and then you can spit or do whatever it is you want to do with it. So it's only when the food is in the oral cavity that you have voluntary control. Once the bolus goes into the pharyngeal cavity, it will, and into the esophageal cavity, it will progress, and you have no voluntary control over it unless you vomit. Okay, so as we said, when you swallow, you put your teeth together, press the food against the hard palate. That's what forms the shape of the bolus. The bolus then is directed, it follows the vault of the hard palate and is projected backwards. And remember, we said the esophagus is between the trachea and the vertebral bone and the vertebral column. So it will be directed, and it will fly over the epiglottis and end up into the esophagus. Now, at that point, when the epiglottis closes the airway, your teeth will touch. Your top teeth, your maxillary teeth and your mandibular teeth will come into contact. At that point, you will hold your breath. You have to hold your breath because the epiglottis has closed the airway. And then once the bolus goes over the epiglottis springs back open again, and air can now flow again, which is why now if you're laughing and if you're distracted, it is very easy to choke. Now, the rapid contraction and relaxation takes place in under a minute, okay? And it's necessary to have this reflex coordination, because there are very many muscles that are involved. Now, there's one thing that can go wrong. Okay, as we said, when you're chewing, you can have people having problems in the mouth, one, because they cannot chew. Well, definitely, if you've got a broken jaw, you cannot eat. But even if your jaw was not broken, if you've got caries teeth, missing teeth, then mechanical breakdown will be inefficient. If you have pain in your teeth, they may be all there, but they're painful, or for some reason or another. So those are the kind of things that would interrupt. Of course, with children who are born with a cleft lip or cleft palate, then food, instead of going into the pharynx, goes into the nasal cavity, and you can cause lots of infections. There's very many and choking, which those asphyxiation, those are the problems that you would get. When it comes to swallowing, infantile swallowing is okay. When a baby is born, they suckle, they live on liquid, and the milk is coming from their mother's breast. They have no teeth. So the teat of the breast is the one that goes into the oral cavity. The lips form a seal against the areolar part of the breast. And because now you have that seal, when the baby now lowers the mandible, you create a vacuum in the mouth, and the mouth, that plus the let down reflex of the mother, is what is going to propel the milk from the breast into the oral cavity. Now, with the jaw lowered, the tongue will tend to be in a concave shape. So the milk is going to pool in the hollow of the tongue. Then the baby will raise the tongue, and the tip, there'll be an incline, and the milk will flow to the root of the tongue and then pour over the esophagus, over the epiglottis into the oral cavity. So basically, swallowing fluids, like the baby when they're suckling, is different from when we are chewing. Because when we chew food and then we swallow, this is now what we call the adult swallow, the tip of the tongue presses against the anterior teeth, okay, the lingual aspect of the anterior teeth. And the tip of the tongue presses there. The sides of the tongue, the lateral borders, are going to press against the posterior teeth so that the food is completely contained between the tongue and the hard palate. Then you get a wave of contractions, which involves the intrinsic muscles of the tongue. Yeah, the longitudinal, the vertical, and the transverse muscles of the tongue. And the wave of contraction starts from the tip of the tongue, and it will push the bolus. The bolus will then push against the vault, or the highest part of the palate, and it will be propelled along that hard part till it goes to the what's it called? Till it goes into the pharynx and is directed now over the epiglottis into the esophagus. Now, when a baby doesn't have teeth, or when a baby is newborn and they're suckling, one, they're swallowing with their teeth together. Okay, adults swallow with their teeth together, because remember we said your teeth temporarily come into contact, you cease breathing because the epiglottis is closing the airway, then the bolus goes over and then you open. So every time you swallow, you raise your jaw and your teeth come into contact ever so slightly, and then they open again. A child, an infant, a newborn has no teeth. So even if they were not suckling, even if you were feeding them with a spoon, what would happen is that they can't put their jaws together because they have no teeth, and the gum pads are built differently. So what I'm trying to tell you is that babies, when they're suckling, they actually learn to swallow with their jaws slightly apart. Now, I would not recommend you do that because you guys will choke, especially if you're eating groundnuts and and popcorn and things like that. So do not go and try this at home. But once the teeth erupt, then the child has got to learn to change and to be able, because now they don't have anything in the mouth anymore, which is why pacifiers are not very good, because the child doesn't learn to press the tongue against the teeth so that the wave of contraction can then go backwards distally into the mouth, starting from the tip of the tongue. So what happens in infantile swallow is the child swallows the way they've been swallowing when they had the mother's teeth in the mouth, And because the tongue is such a powerful muscle, when they swallow and the tongue moves, it is going to stick out of the oral cavity. And if the teeth are not together, then the tongue is not contained within the oral cavity. And when the tongue is contained within the oral cavity, the tongue is pressing against the teeth from the inside, but outside you've got your lips. And if your lips are closed, then your teeth are not going to procline and stick out of the mouth. But if you have this movement of the tongue, and we swallow about, they say about 300 times, is it 300 or 600 times a day? So if you swallow and your lips are not touching to contain the teeth inside, what will happen is that the pressure from the tongue is going to push the teeth outside, and then you'll start, what do you say? It will be unsightly, it will be anesthetic, it will not be very pleasant when they put their teeth together, then their back teeth together, their anterior teeth don't touch. Now, let me just show you, because I have a picture for that. So this just shows you the bolus as it's traveling from the oral cavity through the pharyngeal cavity. And after this picture on the right-hand side, it will be completely in the esophagus, in which case now it will travel down, helped by gravity, to get into the stomach. Remember also that the lower esophageal sphincter is not an anatomical sphincter. And at that point, the esophagus is going through the diaphragm, an opening in the diaphragm. And when you're breathing, the diaphragm, when it's tight, it contracts around the esophagus, and so helps to prevent reflux of food from the stomach back into the esophagus. Okay, so this shows you now the anterior seals formed by the lips against the mother's breast. And you can see now here that the jaws are actually apart because the baby has got to hold the mother's teeth in the mouth. So when they're swallowing, they're actually swallowing with the mouth apart. Fortunately, because their face and head and neck region is smaller than the adult, then the distance that the liquid has got to travel is not so far. So chances of them swallowing is choking is not as high as it would be in an adult. This is a child or someone with an anterior open bite. You can see here that they have put their teeth together. Their back teeth are touching, the maxillary and the mandibular teeth. But when you look in the anterior part of the mouth, there's a big space. In fact, okay, the teeth, okay, the canines haven't erupted yet, but still you can see the tongue, and the tongue is between the teeth, and that is what is actually preventing the teeth from meeting. So what we're saying is that you're going to get developmental problems, which will affect even the posture of the face and will make them look bad. So habits are the ones that are going to affect the development of the face, not necessarily always genetic. Sometimes it's a habit that you do. Sometimes people even suck their digits, suck their fingers. And because the fingers are perpetually between the anterior teeth, or even if they put them on the side, then when the face develops and the face is growing, you get a disconnect between the growth of the mandible and the maxilla, and you can still end up with an open bite. Okay. Now, if we go back to the esophagus and the motility, the bolus actually travels through the esophagus at a speed of about 4 centimeters per second. And if you've ever swallowed a very large lump or a large piece of meat, sometimes you swallow and then you feel, even after it's gone down, you can still feel like it's traveling down. That's because of the sensation, the receptors, the pressure that is caused when the bolus is going down. Okay. Like I said earlier in the day, that we have a mixture of skeletal and smooth muscle in the esophagus. So go back to your developmental, the development of the digestive system, and that will explain to you the areas where you'll find weaknesses, because you may have a defect where the skeletal and the smooth muscle don't knit together. So you have areas where you don't have complete communication between this skeletal and the smooth, and that is what would cause your achalasia. Now, in achalasia, it's a developmental problem, and what happens is that you get a failure of the muscle to relax. So it will dilate as the bolus is coming through, as you know, it will dilate that part of the esophagus. But once the bolus has passed, because you don't have complete development of the enteric ganglia, enteric nervous system ganglia, you get a failure of the muscle to relax. That reflex is not there. So it remains patent. And because it can't relax, it can't push the bolus forward. And you get dilatation of the esophagus and very many symptoms associated with that. The other problem that you can get is gastroesophageal reflux diseases. Yeah, it's not one disease, there are very many conditions. And things that would cause a reflux disease. Okay, one of the things that you would complain or they would complain about is a heartburn. The feeling of heartburn is when you get acidic content, vapor, or the hydrochloric acid somewhere going back into, through the lower esophageal sphincter and coming to irritate the mucosa at the bottom of the esophagus because it's acidic. Now, the heartburn is actually referred pain from there, and that's why people described it as a pain in the heart, okay? That's why it's called heartburn, but there's no hearts that are burning here. And gastroesophageal reflux diseases, there are many things that can cause that. It's not just a high production of acid in the stomach or intake of acidic content. In fact, primarily, if you are obese or you eat a lot of food, your stomach will expand. Yeah. In fact, they say the stomach is a bottomless pit. So the more you give it, the more it wants. If you have a heavy meal, and then let's say at 11 o'clock at night, and then you climb into bed to sleep, one, you've confused your body, because in order to sleep, all your systems have to shut down and slow down until you have the basic heart rate and respiratory rate, and basic temperature maintained. But now what you've done is you've eaten a meal, you have loaded your gastrointestinal tract, and you've told it, go full throttle, process the food. Then you climb into bed. First of all, as you become drowsy, even your enteric nerves are going to be affected. So they will slow down. So the food will now pour and go back. And remember, the lower esophageal sphincter is not like the pyloric sphincter, so it can't tighten. And it can pour back into the esophagus. So so obesity, having a very heavy meal before you go to sleep, which is the reason why you're advised to eat at least 2-3 hours before you sleep. So those of you who went to boarding school and you ate dinner at 6, was it 6 or 6:36? By the time you're sleeping after prep, 8, in fact, sometimes you're even hungry because the stomach has emptied and you've used a lot of food. So what I'm saying is that these are the habits that people are seeing now that will have long-term effects and cause derangement later on. So this is just a picture showing you the junction where the esophagus enters into the stomach. So again, the stomach, the relationship between the stomach and the esophagus. So thank goodness for us that the esophagus does not enter into the top of the fundus, because every time the stomach contracted, stomach content would shoot back into the esophagus. So it actually enters at an angle. You have your lower esophageal sphincter. Remember, it's going penetrating through the circular muscle of the diaphragm, which helps also to contain the contents into the abdominal cavity rather than allowing them to come back to get the reflux disease. In the stomach, remember we said it acted as a blender, and we said that the stomach is the most highly muscular part of the digestive system. You have the outer longitudinal, inner circular layer, and finally you have the innermost oblique. And because of that, the stomach is able to perform mechanical digestion. So what we said is that for those of you who don't chew your food adequately, then your stomach does the job. So what we call adequate chewing, how many chews are you supposed to perform? I have heard all numbers from 32 being the lowest. And I don't know whether people think 32 because you have 32 teeth, I don't know. But for a fact, science has shown the more you chew, the better your digestive system, your stomach is able to perform its job, because the more you chew, the more you stimulate saliva secretion. When you chew, the more fibrous the food is, the more saliva is going to be produced from the parotid gland, which is going to wet the food better. It's going to help initiate digestion in the mouth. And when the food gets into the stomach, it's no longer dry and highly particulate, so that the stomach is able to mix it now with the gastric juices more and make chyme, so that gastric emptying is now the most important thing, because that's where you're going to release the contents of the stomach into the duodenum. And already we said this is a critical phase during the digestive system function, because the highly acidic chyme has got to be released into the duodenum at a rate at which the duodenum can neutralize the acid so that the enzymes can be able to perform their work. Now, there's something that is known as receptive relaxation, because I'm sure you've realized you can eat food, especially if you're really enjoying yourself and the food is really tasty. You can eat and know that you're full, but if you were brought a little more, you would still be able to eat and swallow. So how does your stomach accommodate that? Yeah. Now, receptive relaxation is actually mediated by the vagus nerve. And what it does is that you stimulate the vagus nerve and it causes relaxation of the stomach so that it can accommodate an extra amount of content without increasing in pressure. So that's what is important about receptive relaxation, that you get a vaguely mediated relaxation of the stomach muscle so that it can accommodate more content without necessarily building up pressure, because the stomach has already got contents in it. Now, as we said, gastric emptying is critical to gastrointestinal function, because, as we said, the acids are going to destroy the duodenal mucosa, so you have to release the gastric chyme in small quantities. Now, of the three basic food types, the carbohydrate, protein, and fat, fat is the most difficult to digest. It takes longer, okay? Carbohydrate is probably the easiest, the quickest, followed by protein, and then followed by fat. So if you eat a food or a meal that is high in fat, that fat, as it is released into the duodenum, is going to delay gastric emptying, because the acid, as we said, if the food is highly acidic, that will also delay gastric emptying through the mechanism we talked about, the reflexes, the duodenogastric. But the amino acid, the acid is because they need to be neutralized so that the enzymes can work. The fat has to be released at a rate so that will not overwhelm the bile, which has to emulsify the fat, because remember, fat and water don't mix. So carbohydrates and proteins are relatively water soluble. They will mix with the gastric juices and life goes on. But the fat has got to be emulsified by the bile so that it can be mixed in this aqueous medium, and also so that the enzymes can be able to work as the fat droplets are broken into smaller and smaller pieces. So gastric emptying is critical, and gastric emptying is actually regulated from the stomach. Even though you've got input from the reflexes, as we said, the duodenogastric, it is the inhibitory reflexes that are going to reduce the rate at which gastric chyme enters into the stomach. So if you look at this, this is just a diagram to show you that churning mixture. And I'm sure today, if you Google, you can actually get maybe a little YouTube video that will show you maybe even in real time how the stomach works and how it grinds, because we can actually put fiber optic fibers into the mouth, a scope, an endoscope, which can help you to see that. So this is just a diagram to show that. I've forgotten which book this was from. Okay. Now, in the small intestine, chyme takes quite a while. It takes about 2 to 4 hours to cross the entire 5 meters of the small intestine. The segmental contractions we said were critical here because they delay the transit time of the luminal contents, and they help enhance the mixing of the luminal contents with the intestinal secretions. And we said that most of this, you'll be able to see those contractions in the practical, and the interstitial cells of Canal are the ones that are going to determine the rate at which the basic electrical rhythm is determined here. So even after severing the piece of gut from the animal, disconnecting it from the central nervous system, from the sympathetic, parasympathetics, from the blood flow, you will still see the basic enteric basic electrical rhythm. The other thing that I wanted to point out, or that you need to know, is that agents of analgesia and anesthesia will affect motility. Remember, the enteric nerves are fine, non-myelinated nerves, which means that even if you give somebody anesthesia or even analgesia, they're the ones that are going to be affected first before the larger myelinated nerves. This is the point of, well, the reason why we don't feed people before theater unless it's an emergency, a surgical emergency. If it is what we call elective surgery, something that you're planning for, you normally starve them for at least 6 hours or more, so that by the time they're being put under anesthesia, the stomach is empty, so that there'll be no need, you'll not be stagnating things in the digestive system for that long. You won't get putrefaction. And also, it will also prevent the person from, because if you're putting, when you're anesthetizing or if you're intubating, they will feel like throwing up, and sometimes if their stomach has got content, then they can asphyxiate. So those are the pre-operative preparations that we make. And the other thing is that when the person comes around after the surgical procedure, when they're now recovering and coming back from the anesthesia, you've got to remember that the rest of the body might come back to normal, but the gastrointestinal tract is going to be one of the last ones to come back to normal. And you should not give them food until peristalsis has re-established in the digestive tract. Otherwise, what will happen is that they'll get colic-like pains. The food will go in, it will distend, but there'll be no reflex relaxation, because remember, the nerves are still anesthetized and they can get pain. So that's just some applied aspects of peristalsis. The muscularis mucosa, remember we said it's made up of inner circular layer and also an outer circular layer, and it contracts irregularly, not as regularly as the main muscle coat, but it does contract irregularly, and it allows the mucus lining, the mucosa, to be able to go into, as it contracts, the villi will be pushed into and out of the wall. You get movement of the villi. So it's like straightening your fingers and then curling them. So that's what happens, so that the luminal content in the center of the lumen will be able to be mixed and to come into direct contact with the intestinal cells, so that digestion and absorption can take place. Also, the intestinal villi, as they contract, they change their length. Okay, as we said, they protrude deeper into the lumen and go back as they get longer and shorter. And this is what is going to assist in lymph flow. And remember that the digestive end products of fat are going to be directed in the lymph, will be carried by the lymph and circulated. They don't go via the hepatic portal vein, via the liver. Okay.
Dr. Moretti.
Yes.
It's almost time.
Wow. Okay. Fine. So let me do this. Let me share this with you. Or tomorrow we're meeting at 11. So I think we can finish this. Yeah. And then we can just talk a short note about the secretions of the gut. And then that's it for this year. Okay. Thank you for alerting me to the time.
Okay, no problem. Thank you.
Okay, so you'll share the link, or is this going to be the same one?
I'll share a different one by email.
Thank you very much. Okay.